the pharmaceutical industry is in a kind of crisis at the moment, in the sense that its productivity has declined very sharply in the last decade or so. Its R&D budget has doubled, the number of new drugs has worse than halved.
Something like 40% of the new compounds the drug industry develops have side effects on the heart. It's a huge problem, and one that's costing billions and costing lives. Current safety testing focuses on a drug's action on an ion channel called hERG. Neither the electrocardiogram on its own nor an action on the ion channel hERG on its own, neither of those are reliable indicators of whether or not there will be problems, and neither the regulators nor the pharmaceutical companies are particularly happy with this safety test, but it's the best that they could come up with. And we've been able to show that if you take a more complicated view of the situation, look at the actions of drugs on several proteins at once, then you can get a better prediction. When you've got a very complicated set of interactions with a number of factors involved, you've got to try to understand it. And modelling is another way of creating hypotheses, but instead of doing it as it were out of your head without any mathematics, we do it with mathematics. We do it quantitatively. And so what we've done is to divide the work up into various components. There are people working on the cell models. There are people working on tissue and organ models. And there are people working on the virtual research environment that we're developing, which would enable other people to use the models themselves with a fairly user-friendly interface. And of course there are the experimentalists who are interacting with us in various parts of the world.
As a mathematician working with computers, I try to take information about how people think biological systems work, simulate that, and then see if that's how things really work by trying to make new predictions. The side effect that we're trying to predict is called torsade de pointes, and this is a life-threatening arrhythmia that can arise because you've been taking a drug for perhaps for 6 months, and then perhaps only in 1 in 10,000 people. And so it's very difficult, notoriously difficult, to predict whether this is going to be a side effect of a drug or not. We did a study with experimental data on 31 drugs, and this was for the block on 3 channels, the fast sodium, L-type calcium and the hERG potassium channel. And commonly only the hERG potassium channel is screened, so we wanted to see if more information on the torsade de pointe risk could be gleaned from the 3-channel block rather than just the 1-channel block. We found that in our study, using the existing measure, then 9 of the 31 drugs were severely misclassified to the wrong end of the risk scale. Safe drugs were said to be dangerous and dangerous drugs were said to be safe. Using our new simulated measure, only 1 drug was misclassified instead of 9. We think this tool will scale up to a larger number of drugs, so one of the studies that's happening at the moment is that GSK are measuring for 60 other drugs in order to validate this tool, but we think it will be at least twice as good as the existing measure.
Here at the UPV, we are working in 2 areas. We are developing models of drugs at the ionic level and at the cellular level, and we are also using the sensitivity analysis in order to improve existing models. In this project, we have developed computational models of dofetilide, lidocaine, and cisapride. These drugs are already in the market. However, they can cause the negative effects Gary Mirams is looking at. By modeling how these drugs work, we hope to learn what divides safe drugs from those that cause negative changes in our heart. In my part of the work, together with our colleagues on Oxford, we have developed a sensitivity analysis software. This can test every parameter of the model in order to know which ones are important for the output of the simulation. As far as we know, we are the only group in the world that is systematically analyzing the effect of ion channels parameters at different cardiac electrophysiological levels. We do this because within this project, we have to link our drug models together with cellular models to run tissue simulations, whole hair simulations, and even ECG. So we have to be sure that they behave properly in every scenario.
So what we are trying to do is to integrate all the knowledge we have from other sources in how drugs interact with the heart. This type of research involves a coordination between physiologists, engineers, mathematicians, that only by putting together a group of people with the right expertise you can obtain this type of models. In order to simulate the electrical activity of the heart, you need a description of its anatomy and a description of how it functions electrically. So the fiber architecture determines how the cardiac fibers are oriented in the heart. This is the first model that incorporates a full description of this specialized conduction system. So we have points in the mesh where we solve certain equations to simulate the electrical activity of the heart. Building on the models we have developed, we can now change parameters in the models and properties in the models to be able to investigate different scenarios like female versus male, animals or humans, and also the presence of disease conditions, or some disease conditions that are better characterized in the experiments or in clinical data, like ischemia or heart failure. I think the next step is to bring these methods to clinical scenarios or industrial scenarios, and for that we very much need to validate the models properly and understand better what they can bring in terms of predictive power.
So we started with the goal of trying to solve these problems in as near to real time as possible. And at the start of the project, that would have taken several weeks to solve, even on the most powerful computer. Now, we haven't got to exactly real time, but at the moment, if we solve the same type of problem that we're trying to solve at the start of the project, we've gone from several weeks to 10 seconds for that type of problem. There are all sorts of techniques that you can use to make these codes go faster, and we've explored lots of those techniques. The most obvious one is just to use more processors. But there's a problem with that because each of those processors have to talk to one another, and eventually that slows the process down. So there's a technique called adaptive meshing. What happens with a heartbeat is that electrical activity passes through the heart in a wave. And when we're trying to solve these equations, we're trying to solve the equations where things are changing very rapidly, so as that wave passes through. But as the heart relaxes back after the wave passes through, there's not as much activity, and you can actually solve the equations on what we call a much coarser mesh. The models that I talk about are the mathematical models, mathematical equations. Now, when we implement the numerical algorithms that solve those equations, we do that in software. And we've developed a large software package which has around 300,000 lines of code now, called CHASTE.
CHASTE is very important. It's the only open source software. But in order to have the tool accepted, you also need to show that it's useful.
It's very important that the software be usable. And one of the big aims of the project is to ensure that the virtual research environment that we're creating is one that is totally usable by the pharmaceutical industry, by the regulators, and of course by future researchers.
If you want an application that can repeat an experiment that was done 5 years ago, then yes, you can have that at the command line, but you also need to understand all the command line options available to you. You need to understand all the protocols to get data from different places, and at the end of it, all you have is your results on your local machine. The intention of the VRE is to reach out beyond those dedicated researchers to perhaps people who are interested in what's being done. So we've decided to create a web interface to a lot of the tools that have been created, just to allow people to try things out without needing to install a lot of software on their system. So that graphically you could have a presentation of the simulation data against the equivalent publication data compared to, for example, data that was derived from experimental means.
This database is important because results published in scientific papers are not very well structured. I mean, each researcher published results in his own way, and thus it is quite difficult to retrieve and compare results coming from all these different publications. As simulation results are more or less structured like experimental one, it's quite easy to store results of simulation studies in the same database. So later on it will be quite easy to compare a real-life experiment with simulation results obtained in a virtual world.
Europe has a huge opportunity now because it's built up an almost unrivaled expertise in this area. It seems to me that we've had a huge degree of success, as evidenced by the extent to which the pharmaceutical industry and the regulators are interested in what we're doing and using what we are doing. I think we could get even better success than we've got at the moment, because even with looking, for example, to take one of the projects, even with looking at the actions on 3 of the protein components in cells, we can get a much better prediction of drug activity than any of the other criteria used at the present time. If we could do that for 5, 6 or more mechanisms and encourage the drug industries to interact with us to develop in that direction, we will progressively get to bigger and bigger confidence in the way in which modelling can be used to help the industry.